Hello everyone, welcome to another video on enzymes by educators. Everything that happens in a cell growth, such as uh, reproduction, digestion, interaction with the environment, response to a stimuli, etc., happens because of a chemical reaction, right? And what is this chemical reaction? This chemical reaction takes place or happens because there is a change in the atoms or the molecules which yields different substances which forms different structures. So either it involves some breaking of bonds or it involves some formation of new bonds. So some old bonds is either broken or some new bonds are formed. So this is basically the chemical reaction. So this is what a chemical reaction is. Now all this chemical reaction, to kick start this chemical reaction, we need a catalyst. What is this catalyst? A catalyst is substance which speed up this chemical reaction, and without changing the reaction uh, type or the state of the reaction, it just kickstart the chemical reaction. So catalyst is basically something which kickstart the uh, chemical reaction. Now what is this enzyme? This enzyme is a typical catalyst. It is a biochemical catalyst which help in the start or kick-starting any kind of reaction in the body. So let us learn what is an enzyme, what are the different functions of enzyme, how enzyme work, and how enzymes are named in our body. Welcome to Educators, myself Manjishta Chatterjee. Don't forget to like, share and subscribe my channel. So enzymes are protein in nature and they are biocatalyst or biological catalyst, as I've told you, as I've mentioned. These accelerate the chemical reactions or make the chemical reaction fast, and they also act upon some molecule which is known as the substrate, and they convert the substrate into some new product or they form some new product which is known as the products. So they convert the substrate into some different molecules altogether, which is known as the product. So enzymes are biocatalysts which acts upon the substrate and they form the products. Let us see this graph. What we can see, with enzyme, the activation energy is this much, and without enzyme the same work is done with this much of activation energy. So whenever a reaction is done with enzyme or without enzyme, that particular reaction will be done or will be performed in our body and we will definitely receive the products. But with enzyme, the time will be or the activation energy will be much lesser than that of without enzyme. So enzyme definitely help in accelerating the process of the reaction. Now for that, for accelerating, we need to be in contact with one another. The reactants like CO2 and H2 over here, they have to be in contact with each other. The bonds must be broken, formed or rearranged, and these reactions, they need the input of the energy. And as I've mentioned, an activation energy is needed or required for the reaction to this to occur. Now how the enzyme act? Over here, how a product is formed, how the enzyme substrate complex be ready. So what happens? These are the two substrate, the red one and the green one. So substrate, the enzyme, they have an active site. Now, why is this active site? Because this active site are made so that they can easily fit or embrace the substrates into them, and they are held weakly by the hydrogen bonds or ionic bonds. When the active sites hold the substrates, they lower the enzyme activity and they speed up the reaction by substrate orientation, by stressing the substrate and stabilizing the transition state, and by providing a definitely microenvironment, and by participating directly into the catalytic reaction. So by doing all these four types of function, the active site of the substrate, they start working or they start catalyzing. And what they are up to, they form or change the substrate into two new substrate, which or two new complex or two new molecules, which we call the products, and these products are released. Once these products are released or the substrate are converted to these products, the active site is again become available for the two new substrate mole. So this is how the whole thing continues. I'll repeat it again. So first the active site becomes empty, then is it is induced, like it is such actively participates for the substrate to come and bound to it with weaker bonds like the ionic bonds and the hydrogen bonds. Once the hydrogen bonds and the ionic bonds mix with the substrate, they lower the enzyme activity and increase the catalytic reaction by all these functions, by making the microenvironment favorable, by properly orienting the substrate, by proper catalytic reaction, and the substrate are converted into products. The products are released thereafter, and then again the enzymes are ready for new substrate. So this is how the enzyme acts or the enzymes help in catalytic reactions. Now, what are the characteristics of enzyme? Very important. As I've mentioned, enzymes are protein in nature. All enzymes are not like all enzymes are protein in nature, but all proteins are not enzymes. You have to know that and mention that. And always remember that enzymes are character substrate specific, so they act on the specific substrate meant for them, and always they act at a particular temperature and a particular pH. So few loves acting in acidic pH, few loves acting in basic pH, few loves acting in neutral pH. The enzymes have a protein part which is known as the apoenzyme, and a non-protein part which is known as the coenzyme, which bind together to form the whole enzyme, which is known as the holoenzyme, to which the substrate come and bind. So one is the protein part, another one is the non-protein part. Protein part is known as the apoenzyme, non-protein part is known as the coenzyme, and this substrate then bind with the total protein and non-protein, that is the holoenzyme part or the whole enzyme part. Some enzymes are zymogen, that means they are inactive in nature and they needs to be activated. Sometimes their autocatalysis activation is done. After they are active, they can actually change the other and do the catalytic reaction. So let us learn the different apoenzymes, the different cofactors, and also the different zymogens. So like this, pepsinogen is, we have learned in the digestion chapter, pepsinogen is a very important zymogen whose active form is pepsin. Similarly, trypsinogen, trypsin, prothrombin, thrombin. Over here, some activators like the cofactors and coenzymes are there, which are the non-protein part in an enzyme. Now, some magnesium, potassium, calcium, zinc, um, iron and copper, they bind with the enzyme molecules. These inorganic compounds and ions are known as the cofactors. And some non-protein organic molecules, these are the inorganic compounds, are known as the cofactors, and the organic factors are known as the coenzymes, like the NAD, FAD, coenzyme A. So they are all activators and help enzyme to do their function. Now, enzyme nomenclature is very important. Different enzyme has different kinds of name. How can we name the enzyme? Now, sometimes on the basis of the substrate, we add the suffix like urea, urease, arginine, arginase. That means it acts on the substrate specific. As I have so told that enzymes are basically substrate specific. So, arginine, arginase. Okay. Lactose, lactase. Lactase enzyme acts on lactose. Trypsin, maltase acts on maltose, amylase acts on amylose. So these are the different kinds of enzyme which are substrate specific. Some old traditional names are there like the ptyalin, which is the salivary amylase, the renin, pepsin, trypsin. This is all some old name which has been given. Some are denoted as lytic based on their splitting function like proteolytic, lipolytic. Those who help in the splitting action, they are named such. Some are catalytic functions are there, similar chemical reactions are there. Some dehydrogenases are there, oxidases, proteinases, and lipases are there, and some are named on the species of origin, like the the enzyme present in papaya is papain, the enzyme present in ficus is ficin, capsicum is capsaicin. So this is how the enzyme nomenclature is given. So all enzymes are protein in nature, basically key point, and they are substrate specific. They act in a specific pH and a temperature, and they are biocatalyst or biological catalyst. So these are the main key points of enzyme. And while we have studied the digestion and absorption of enzymes, I hope you have learned a lot about the different examples of enzymes and their function. So more when we read about the different metabolism, you will be also knowing about the action of the different coenzymes and the cofactors along with the functions of the enzyme. So I hope you have understood what is enzyme and what the functions of enzyme. I hope it was clear. That's all for today. Thank you. Stay tuned with educators.